It's going, and we can hit records now. Right, apologies if any of you can hear noise in the background. I'm on support for one other workshop, and there are other conferences going on in the same building as well, so there's likely to be a lot of noise. Right, should we get started then?
Sure.
Right. Hello, everyone. Welcome to BGA24. This is session 17 on BUSCO from QC to gene prediction and phylogenomics. And this is with Matthew Berkeley. Please take it away, Matthew.
Sure. So I'm going to share a screen now so I can get going. Hello, everybody. Thank you to Damon and the organizers for organizing this event. So actually, maybe before we get going, we can just go to the repository and start the Git pod so that I can just set up while I'm doing the introduction. So the BGA Academy, you got the BUSCO repository, and then down here you got the opening Git pod. So you can just open that and set it running while I'm talking, doing a few introductory slides, and then it'll be ready to go when we come back to it. And if you need to set up your Git pod, if this is the first talk you've been attending, I think there's a few hoops you have to jump through and sign in through GitHub and all that kind of thing. So it'll just take a few minutes to set up. But I'll do the introduction, and then by the time we get to the first exercise, then it should be all ready to go. So BUSCO, this is an interactive guide. We'll be going through what BUSCO is, how you can use it, how the software pipeline works, the data sets that go along with BUSCO, and then a couple of use cases. I'll be talking briefly about them as well, how you can use BUSCO for things like phylogenetic analysis and other companion pipelines that we've developed to go along with the main BUSCO software. So first of all, about me, my background is not actually in biology. It's in physics, astrophysics. So I did my PhD in 2018 at the Catholic University of America in the United States, doing research with the cosmology division at NASA. So that was a lot of astrophysical image processing. And then I made the move back to Europe, where I've been working with the Swiss Institute of Bioinformatics since 2018 on primarily sequence genomic data. And so since around about the start of 2019, I've been the lead developer for BUSCO, and it's gone through several different evolutions since then. So I'll talk briefly about orthology, which is the science underpinning BUSCO and how it works. So let's move this bar out of the way. Okay, so I can see what I'm. So orthology is, as a science, it's a relative concept. Say you've got a number of different species. Any group of species has a last common ancestor. And there are genes that have propagated through speciation events. In some cases, genes have been duplicated. Sometimes they've been lost. But any group of genes that can trace their origin back to a single ancestral gene, they form an orthologous group. So that's the key point number one about orthology. In our group in Geneva, we run AutoDB. We've created this and we manage it. Currently, it's in version 12 as of the end of last week. We just made the version 12 public. Version 12 represents a huge increase in the number of in species coverage. So it's the largest database of orthology data across eukaryote, prokaryote viruses. So I mentioned this because this AutoDB underpins all of the BUSCO data, and I'll talk a little bit more about that later. So AutoDB takes a hierarchical approach to gene orthology. So say you've got humans and apes, for example, they have a last common ancestor, say at the primates level. So there's a set of genes, a set of orthologous groups that are present at the primates level. And for example, any genome assembly that you create involving any of these species should have all of these marker genes in them. Similarly, mice and rats, for example, have a different last common ancestor. So at the rodent level, there'd be a different set of orthologous group genes. And so as you go further up the hierarchy, you get to, say, mammalia, higher up here, where all of those come together, and there'll be another set of orthologous genes, orthologous groups. So as you climb higher in this hierarchy, there are typically fewer genes at each level, the higher you climb. And so when you're using BUSCO to assess the completeness of your genome assembly, it's always important to use the highest resolution data set, because that way you get the best information about how complete your genome is. So orthologous groups, over half of orthologous groups at any clade are universal. But in some cases, they have been duplicated. Sometimes they're missing. The ones that we're most interested in are the universal orthologous groups. That means they're present in over 90% of the species in that clade, and they're present as a single copy in over 90% of the species in that clade. The reason for this kind of this region of genes, it's interesting because if they're preserved in a single copy universally in a clade, it points to a specific function that these genes are used for. And if we can use the presence of these genes as a proxy for the completeness of a genome assembly. So that's I'll also say there was a talk last year. I think I mentioned on this slide here. It's covered by a bar. Rob Waterhouse gave the talk in BUSCO last year at BGA. And he spent the first hour talking about the science of orthology. So if you want to dive into that a bit deeper, I recommend you check out that talk. So BUSCO. BUSCO stands for Benchmarking Universal Single Copy Orthologs. It's a spin-off project, I guess, from OrthoDB. It uses the data from OrthoDB to assess the completeness of genome assemblies. So the software will output a score for your assembly, and it'll tell you how many of the marker genes for a given clade are it found complete and single copy. So you want this number to be quite high. It'll also flag if a gene is found, a single copy marker is found in multiple copies. So it'll say that we found this as a duplicated gene. You might want to check out something in your assembly. It'll flag if it's fragmented and flag if it's missing. And so sometimes you might want to, it might depend on what kind of genome you're working with. So if you're working with a diploid assembly, you might expect to get a high number of duplicated genes. But typically we're looking for the highest number of complete and single copy genes. So BUSCO provides a completeness assessment of newly assembled genomes, proteomes, and transcriptomes. Let's see if I can close this down here. I can see that cursor. No, back there. I'm going to close down this because I can't see the full screen. Okay. So it takes an evolutionary-based approach, as I touched upon. That's built on data from OrthoDB. BUSCO was first published in 2015, and since then it has grown considerably, and it's become an integral part of all major genome assembly pipelines. So this shows how BUSCO has grown. So since 2015, we've been the bar chart on the right shows every year how the citation numbers have been increasing. These are the papers we just published, or we just got accepted for nucleic acids research journal. It was just accepted at the end of last week, so there's an updated paper coming out just to talk about the updated version of OrthoDB and the developments in BUSCO as well, particularly about the new upcoming data sets, which I will talk about soon, the version 12 data sets. So there's a user guide online. This is the hyperlink to the user guide. And so for most of this talk, I'll be kind of going through the different sections, I suppose, and doing a few examples of how to run BUSCO. But I recommend you check out this user guide because a lot of the basic questions and how it works are laid out in some detail here. So we'll switch to Gitpod now. And we'll just show how BUSCO is set up. So we should have if you manage to set up Git pod here, I might just give another couple of minutes to set this up, just in case people haven't done it. Or so it's just at the repository, you've got the open Git pod link. It takes maybe about a minute or so to load, maybe 2 minutes. It's not too long. So here we've got in the BUSCO directory, we've got these exercises, and I've set up a few different things that we can go through here. But before we get into that, I'm going to just toggle to the user guide here, which is the BUSCO user guide. And the latest version is version 5.8, so that was released just at the end of last week as well. To install BUSCO, there's 3 different ways that you can install it. There's the Conda installation, which has everything pre-packaged and all the dependencies are included. So to install with Conda, you can follow these commands. You use the channels, Conda Forge and BioConda, and you create an environment. A common question that I get on the GitLab issue board is people having difficulty with problems with imports. And it's usually a problem with environments. So I recommend when you're installing BUSCO with Conda, you create a new environment. And the way that works is when you create an environment, the path information is updated in your shell. So when your shell is trying to find the BUSCO command, it'll go straight to so for example, I'll show you here. This might be very familiar to a lot of you, but just in case there are some people who are new to BUSCO, new to the shell, I'm going to show path command here. And this shows the paths that are traversed by the shell when it's trying to find the different software packages. We're currently in an environment called BUSCO, but if I deactivate that and go to the base environment, and I do the echo path again, the path has changed. So we don't have this path, which is where all the BUSCO dependencies are. It's no longer there. So if I try to run BUSCO here, it shouldn't work. Yeah, BUSCO is not found. So I have to activate the BUSCO environment. And then I should be able to get BUSCO, and it works fine. So these are just things to look out for when you're installing BUSCO, because a common problem is that there might be multiple versions of BUSCO installed in the same environment, or maybe you've got a version installed in your base environment or your system that's cross-talking with your created environment. So these are just common troubleshooting things that you should look out for. We've also got a Docker distribution. You can pull it directly from Docker Hub using this command here, and then run it using the Docker commands where you mount your directories, and then you can follow that with the BUSCO specific commands. So in Gitpod, the way I've set it up is a Conda environment. So we don't have to do any manual installations. It's all there because there's lots of third-party dependencies. And you could install it this way if you want to install all these different things and make sure they all work well with each other, but it would take a long time and it's complicated. So it's much better to use the prepackaged distributions. So we'll go to exercise 1. So we've got here exercises. We're going to go into the exercises directory. And here we've got 5 different exercises that we're going to go through. So exercise 1. Also, I'll just before I start this, in the repository, in the README file, I've got the commands that you need to run the different exercises. So you can copy and paste them if you want instead of typing them in. So what we've got in this directory is a gene set, a bacterial gene set. So just looking inside it, we've got different genes that are amino acid alphabet. So we're going to run BUSCO on this. We're going to use the BUSCO command, use the I flag for the input file. We use the input file there. And then we're going to specify the mode, which is protein mode, because our input file is a protein gene set. So that's the bare minimum that you have to have on the command line. But we're also going to add a lineage data set. We're going to specify bacteria ODB10. And we're going to see how well this performs. Also, we have 4 CPUs, so we're going to specify 4 CPUs. So then we're going to set this running. It shouldn't take too long. And you can see the log output keeps you updated on what's going on with BUSCO. So it's configuring BUSCO with the local environment. So that's saying all the things that are available on the path. There are other ways of doing it as well. I'll talk about them later. You can use a config file. But for today, we're just going to use the path, and I'd recommend just using the local environment for most of your runs. It's running in protein mode. Then it downloads this. It downloads information on the latest versions of the BUSCO data, and then it downloads the latest version of the bacteria dataset file as well. These are all remote on our remote servers, but BUSCO downloads them automatically when you run. So you don't need to worry about having those data sets available in your system. So in protein mode, there's one major step in the pipeline, and that it runs against Hammer. I'll talk a little bit about Hammer and how Hammer is used. In this data set, bacteria, there are 124 marker genes. So that means there are 124 genes that are expected to be in all bacterial genomes. And we'll look inside the data set in a second. But just here's the result before we go to the data set. The result is that 100% of these 124 genes are found in it as complete and single copy. So this is a good genome, but the caveat is that we only test it against bacteria. So it doesn't tell us a huge amount about the quality of the genome. We should be testing against the most high resolution data set if we want to really test the quality of this assembly. But just for showing you how the pipeline works, I chose bacteria. It's quick because there's a smaller number of marker genes. So if we look at the folder now, there's this BUSCO downloads folder. This is where the data sets were downloaded. So we'll look inside, and we've got the file versions. That's all the information about the latest versions of the data sets. But in the lineages file, we've got the bacteria data set. So we look inside the bacteria data set here, and we've got all this information that is the same kind of information is available in all of the data sets. But the heart of the data set is the HMM's profile, HMM's folder here. So what this is a HMM profile for each of the marker genes. HMM profile is basically a kind of statistical representation of a collection of similar genes. So all of the species that went into creating this data set, we got all of the genes for, say, in one cluster. This is one cluster of genes. And all of the genes are statistically analyzed to create a HMM profile. And inside these files, you can see, say, if we look inside it quickly, I won't go into this in detail, but this is just a kind of a probability matrix that shows the transition probabilities from one state to the next for the various amino acids in the sequence. The full details on how Hammer works is a different story altogether. There's a user guide for Hammer. It's an external tool. But what we need to know is that Hammer provides a score for how well the proteins, the genes, the proteins in our input file, how well they match each of these HMM profiles. So that's the data set, and now we're going to look at the output of the run that we did, which is this BUSCO GCF. So if we don't provide an output name, it'll automatically name it with a BUSCO prefix and the name of the input file. So if we go into this data set, or into this folder, we'll see we've got a logs subfolder, and in this logs subfolder are all the standard out and standard error streams from all the external tools that are called. And in this case, there was just Hammer, which was called. So we got the HMM search out log and HMM search error log. These are there just for just in case something goes wrong and you want to explore a little bit further. If one of the external tools complained about something, you can find it in these logs. And then we've got the BUSCO log file, which is the same stuff that was printed to the screen, but there's some extra information as well, like the exact commands that were called. We've also got these short summary files. So we'll have a quick look in this one here, short summary. let's look at the text file. And here we've just got a summary of the results that we saw on the screen, and there's information about which versions of the external tools were used. The same information is available as a JSON file with some extra information about the parameters used for your run, the lineage data set, and the versions. So this could be useful if you're running a pipeline and you want to extract some of these values. Instead of parsing the text file, you can just parse a JSON file, and that's easier to do. So that's the top level of the results folder. And we've got this run bacteria ODB10, which is where all the main results are. So we're going to go in here and look around here. So these short summary files are the same as the ones on the top level. They're just copied, because sometimes you can have multiple runs in a single BUSCO, and it'll just copy the ones that are needed for the top level. So we don't need to worry about them. The missing BUSCO list, well, we scored 100%, so this file is going to be empty. But if there are some missing BUSCOs, they'll be reported here. The full table, this is a useful file to inspect after your runs. So it'll provide the list of all the markers, the status, whether they're complete, whether they're duplicated, fragmented, or missing. And then it'll identify the sequence and the coordinates for the sequence. It'll show the score that Hammer reported, the length of the match, and then some links to OrthoDB, where you can find more information about this particular marker gene, or whichever one you're interested in, along with the description of its function. Here. So this is a very useful output file if you're especially if something doesn't look right, if you've got duplicated markers or you've got some missing ones, you can find out exactly which ones are causing a problem with this file. We've also got the BUSCO sequences folder, this one here, and in here we've got all of the sequences that were predicted. So single copy BUSCO sequences. So for example, this marker here, we'll look at this one, and we got this. This was the protein that matched this BUSCO. So you can do the same for all of the different markers. So these folders, multi-copy and single copy and fragmented, they'll be empty because we got 100% on this run. So that's the basis of the output folder, the basics of the output folder. So now I think we'll go back to the slides. So we did exercise 1. That's a bacterial gene set in protein mode. So I talked briefly about Hammer. It's a biosequence analysis using profile hidden Markov models. It's a statistical model that can be used to model biological sequences. And so these are all the details of how Hammer works. It's the hidden states that give rise to a probability distribution, which we saw on the file, probability matrix, and that can be used to predict sequence continuation. So there are certain codon biases which can be modeled, and it's possible to predict protein domains. So after Hammer reports the scores for each of the markers, BUSCO applies several filters. I'll go through the filters that we apply here. This is stuff that we've added to the software pipeline. So the HMM profile matches must exceed threshold scores and lengths. So these scores and lengths are set in the dataset, file. So I'll show you briefly here if we go back to the downloads folder, let's go downloads. We got lineages, bacteria. In here, we've got length cutoff and scores cutoff. And these contain values for each of the marker genes. So the HMM results have to exceed these thresholds. If they exceed the score and length thresholds, they're considered complete. If they exceed the score threshold but not the length threshold, then they're fragmented. And if it doesn't exceed the score threshold, then the match is discarded. BUSCO then removes any overlapping matches. But there is a feature where if it's possible to save a match by discarding unused exons, then it will do. So if you've got one gene that matches, say there's 4 exons and 3 of the exons match the HMM profile and the other one doesn't. And then there's another gene that matches that matches the same another the same HMM profile, and the overlap is in the region that isn't matched on the first one, then it'll discard the trailing or leading exons to preserve the 2 matches without having any overlap. So after that, it'll remove any duplications. So if a BUSCO marker matches as both complete and fragmented, you remove the fragmented one and just report the complete one. And if a gene matches multiple BUSCO markers, we keep only the highest scoring match for that gene. Then we remove any low scoring multi-copy matches. So if a marker matches multiple input genes, we take the highest scoring one, and then any that score less than 85% of that score are discarded. But if they're within 85%, then they're kept and they're reported as duplicated. So the BUSCO pipelines that we've seen, now there's three run modes. We've got the genome mode, the transcriptome mode, and the protein mode. We've seen the protein mode. That's the simplest and quickest mode when you already have your gene sets. Another interesting mode is the genome mode. But to do this, we need to use gene predictors, because if we're using nucleotide input files, we need to be able to predict the gene sets before we score them against the HMM profiles. Yeah, so used to translate nucleotide input into protein gene sets. But this stuff it can be computationally demanding, especially for large genomes. And new and better tools that do this gene prediction are always being developed. So there's new ones coming out that can give some performance improvements. But each tool has different requirements and approaches on outputs. So configuring these different tools is a non-trivial task. So incorporating new ones into BUSCO takes time, takes a lot of testing, and it's not a straightforward thing. Nevertheless, we do have 3 different pipelines, and I'm going to talk about the 3 of them here. The first one was the one that was there in version 3. So version 3 of BUSCO started with a BLAST alignment. And then the regions that were identified in the BLAST alignment were passed to Augustus. Augustus has very precise gene models and is very good at predicting the exact structure of the genes. So the way it looked in version 3 was this. We had the genome mode with the BLAST followed by Augustus. The transcriptome mode just had the BLAST, and then we had the hammer step for all 3 pipelines, followed by the results. For version 4, we introduced Prodigal, which is a gene predictor that specializes in prokaryotic gene recognition. It does ab initio gene prediction on prokaryotic genomes, and it also provides the option to provide translation tables for clades that use a non-standard genetic code. So then for version 4, we ended up with this kind of structure here where we had 2 genome pipelines. We had one for eukaryota and one for prokaryota. But again, they were all passed to hammer for the final analysis. So now I'm going to go back to Gitpod and do exercise 2, which is a bacterial genome in genome mode, just so we can see this pipeline in action. So you can go to the commands in the README if you want to go straight to the correct directory here. Okay, and here we've got another bacterial genome. This time it's a nucleotide file. So we're going to do BUSCO with the input file here. We'll set the output file to the output folder name to be, say, bacteria genome mode. That's what we'll call the output folder. We set the mode to genome instead of protein like we had last time. And we're going to use the bacteria data set again. And we'll set this number of CPUs to 4. So we'll set that running. And you can see the log is printed to the screen as before. Now, instead of jumping straight to Hammer, we've got a couple of steps before that. We download the data set as before. And then the first step in the genome pipeline, looking up here, is, I can scroll, yeah, BB tools, which is another external tool that provides some statistical information on your assembly, things like your N50 score, and a few other statistical parameters. And we can look at the output of that. We report some of that output in the BUSCO output as well. Then after that, we run Prodigal. And because it's bacteria, we use genetic code 11. And these parameters are all set in the data set files. And BUSCO just reads them. So we run Prodigal, and then we pass the results to Hammer. And again, we got 100% completeness on this, which again is just illustrative. It's not, this doesn't necessarily indicate a good assembly, because you'd have to use a high resolution data set to determine that. But we'll look in, I want to go into the BUSCO downloads folder again, so we look at bacteria. So there's one file we didn't really look at, this dataset.cfg file. So I'm going to look inside that. And so here we've got some information about the data set. We've got the name, we've got the domain, the data was created, so that's kind of a version. So sometimes we update the data sets with some minor things, and then the date will be updated. The number of BUSCOs and the number of species that went into creating this data set. Here we've got the Prodigal genetic code. So this is what Prodigal reads in the prokaryotic data sets. Most clades use 11, but some use 4. And then depending on what clade you're working with, you might have a reason to use another one. But when BUSCO is running it, it'll test 11 first, and then if the scores are not good enough, it'll use 4. It'll do it again and use 4. And choose the optimal result of the two. And it chooses it based on the coding density range. So if you run it with 11 and you run with 4, and one version provides a coding density that's higher than the other, we choose the one that's higher. So these values here are also used as well, but that's the nitty gritty, and we don't really need to get into that right now. So that's the values in the data set. The results here in, let's go back to exercise 2. Where are we? Here we are. So the bacteria genome mode, this is our output folder. The similar kind of structure, we've got the short summary files, and we've got the run bacteria output as well. We've got this Prodigal output now as well, that contains more information about the predicted genes from Prodigal. You can have a look at the output from Prodigal there. FAA, for example. And this stuff is passed then to Hammer. You don't really need to look at these files. These are just internal files. We use this information to pass to Hammer. The most interesting information, I guess, to look at on a first pass is the full table file, which we looked at before. What else did I say I'd show? I wanted to show the BB tools output, which is in bacteria genome mode in the logs folder. You got the BB tools output. I'll look in there, and you see this is the information that BB tools creates. So you got N50 scores, L50 scores, etc., scaffold lengths, maximum contact lengths, and so on. Right, so that's exercise 2. Okay, so after that, okay, one second. We introduced Metagenomics, which is an alternative gene predictor for eukaryota. It's good at eukaryotic metagenomics, particularly. And we decided to introduce this as an alternative to the Augustus pipeline, alternative to the BLAST and Augustus pipeline. Because based on our analysis, it seems to perform quicker than the BLAST-Augustus combination. So here's an example of the runtime. You can get an improvement in the runtime using Metagenomics. Though it's less precise as a gene predictor than Augustus. So that was something to take into consideration. It's possible to pass parameters to Metagenomics through BUSCO, and I'll show you how to do that shortly. So you can adjust the sensitivity value for Metagenomics, and you can increase the runtime even further without having too much of a drop off in the number of complete BUSCOs detected. So now after adding Metagenomics, we had another pipeline here in genome mode. We have the Metagenomics pipeline and the BLAST and Augustus pipeline. And we also added Metagenomics for the transcriptome mode for eukaryota. And we kept the BLAST for prokaryota. So then a couple of years ago, MiniProt was released. And this is a very fast aligner. And we have incorporated a MiniProt pipeline as well into BUSCO. So now we've got MiniProt here alongside Metagenomics. MiniProt is now the default pipeline. It's faster than both Metagenomics and Augustus, though care should be taken with the output because it's an aligner. It's not a gene predictor. So sometimes the output predictions that it creates can contain internal stop codons, and they're not always reliable as genes for subsequent analysis. We would recommend if you're using the BUSCO output for subsequent analysis, we recommend using the Augustus pipeline because that has the most precise gene models. Exercise 3, we're going to do some eukaryotic data now. We'll go back to Gitpod here. Copy the links here for exercise 3. So now there's nothing in this data set yet because what I want to do is take the sample data that we've got on the in the BUSCO repository. So this is the BUSCO repository on GitLab, and we've got some test data here. We're going to use this data for this exercise. So first of all, we're going to clone the repository, and then in here we've got a test data, and we're going to take the eukaryotic test data, the genome here. It's just a partial genome because I don't want to spend too long running genome pipelines here because they do take a lot longer than the bacterial genomes. So here we're going to just copy the genome into our directory. So copy test data, eukaryotic genome into that directory here. So it's here in exercise 3, we've got genome here. And the command that we're going to do is BUSCO again. The input file is going to be genome. The output file will say MiniProt genome pipeline, because MiniProt is the default. We're going to use the eukaryota data set as the lineage. And we're going to, what else haven't I done? Set the CPUs to 4. Is there anything else that I haven't included here? No, that's all we need for this. So we run this. I missed something. Oh, I forgot the mode. Of course, I forgot the mode. Okay, so I'm going to, where is this now? I'll remove that BUSCO log file. Add the mode, which is genome mode. Okay, now it should work. So here we've got the same log output again. We've got this time we download the eukaryota dataset. We run bbtools again to get some statistics on the input file, and then we run MiniProt. MiniProt builds an index first, and then it does an aligning step. This takes about a minute. So I'll just wait for this. So after the alignment step is completed, there's a filtering step that BUSCO applies because there's a huge amount of alignment data that's created by MiniProt. And especially for large genomes, it's suboptimal to pass all of the data onto Hammer. So we do some filtering at that stage, and we reduce some overlapping regions, that kind of thing, before passing to Hammer, and then it runs quicker at the Hammer stage. So here we've got, there's 125 markers now at the eukaryota dataset level. And here we've got a 19.2% complete genome. So again, it's just for illustrative purposes because it's a partial genome. It's not an actual genome that we're working with. So here we'll look at the output folder here. Yeah, I think we'll just go straight into the output folder. So mini-prot genome pipeline. Well, actually, before we do that, go back to this. While we're before we go into the output folder, we're going to run the same command again, but this time we're going to use the Metagenomics pipeline. So it's the same command, but we're just going to use this Metagenomics flag at the end. And that'll run the Metagenomics pipeline instead of the mini-prot pipeline. So that shouldn't take too long again. So an interesting thing about the Metagenomics pipeline and the Augustus pipeline that is not the case in the mini-prot pipeline is that once the hammer stage is completed, any BUSCOs that are missing or fragmented are isolated, and the pipeline runs again with an expanded set of reference genes. So it goes through the Metagenomics stage again, and then hammer again to try and cast a wider net to find BUSCO matches. So that's why you'll see two blocks of HMM search commands here. So here we've got the same score, it turns out 19.2%. And then finally, we're going to look at this one here, which is the Augustus pipeline. So here we got the Augustus flag at the end, before we had the Metagenomics flag at the end. And if you don't do anything, then it just runs mini-prot by default. So we've got eukaryote already downloaded, so we don't need to download it again. It runs bbtools as before. It creates a BLAST database. And then by default, the top 3 regions for each marker are passed on to Augustus to predict the gene more accurately. And that parameter can be adjusted as well at the BUSCO command line, and we'll talk about that in a little bit. So here there's 52 tasks passed to Augustus. So there's it looks like there's 52 regions identified by BLAST. So it's running those on Augustus. And again, you'll see a repeated step here where it retrains Augustus based on what it's found and tries to find an expanded, it tries to find more markers based on an expanded reference set. So here we got the output, which is 18.4%. It's a little bit different. So there are numerous different reasons why the pipelines can score differently. But the general guiding principle is that for accurate gene predictions, you should use Augustus. If you're trying to assess completeness, and that's all that really matters to you, then you can use a faster pipeline, use mini-prot, or use Metagenomics if you'd like. Okay, so here we've got the three output files here. And we can look in to see, for example, looking at the full table of, say, the Augustus one. We've got the full table file here. We can see which ones are complete, which ones are missing. And then you can do the same with, say, the mini-prot output and see which ones are complete and missing. You can compare these if you'd like, if you want to get into it and see which ones are the same, which ones are different. We won't get into that now, but I'm just showing you the 3 different pipelines and how they work. Okay, so next we've got BUSCO for Metagenomics. This is a feature that we added in version 4 a few years ago now. Well, what is metagenomics? Basically it's sampling an environment and sequencing everything instead of a single sample, a single genome. The resulting sequence assemblies may contain unknown organisms, and with version 4, we introduced the auto-select lineage pipeline. So there are three phylogenetic domains. There's archaea, bacteria, and eukaryota, and each of these has a parent data set in BUSCO. So what we do is we run against all three of these parent data sets to identify which domain scores best. And then based on the markers that are found, we try to place those markers on a phylogenetic tree using SEP. And then we identify the best possible data set and we run again. I'll show you a little bit. There's a better diagram coming up. So BUSCO is the only tool able to evaluate both microbial eukaryotes and prokaryotes. And we have also introduced viral data sets. There's 27 viral data sets that are available. So SEP, it's been around for quite a while, 2011. We use this to place the markers onto a phylogenetic tree that we also provide. It's provided along with the data sets. And it'll identify which data set is the optimal data set to use. So here's the diagram. So for example, if let's move this bar out of the way. Again. Say you've got an input and you're not sure what is in it, it'll run on all three of the parent data sets, archaea, bacteria, and eukaryota. If it's archaea or bacteria, the data set will say that the domain is prokaryota, and so it'll run prodigal. If it's eukaryota, the data set will say it's eukaryota, so it'll run the default mini-prot, or if you requested Metaeuc or Augustus, it'll run them instead. Then it'll score them after the hammer step. And then based on which of the parent data sets scores highest, it'll take those markers and try and place them on a tree using SEP. And in this example, say Diptera is chosen as the optimal data set. And so that goes back to the top and you run again using the Diptera data set, which will trigger the eukaryota pipeline. And you get your final results at the end. So we'll do a quick exercise using auto-lineage mode. We'll use the bacterial genome, I think, from the second exercise. I think it's the same one. Yeah, so we've got the genome already in exercise 4, the subfolder. So you can copy the directory here and go to the subfolder. And here the command is BUSCO with the input file name. We use the output directory name. We can call it auto-lineage pipeline or whatever you'd like to call it. It doesn't have to be that. Set the mode to genome, the number of CPUs to 4, and then we use the auto-lineage command here. So we'll type that out here. Well, we'll just copy it. It's quicker. Okay, so I'll run the auto-lineage pipeline and we'll see what happens here. We've got, it says starting auto-select lineage because we didn't specify a lineage dataset folder. So it starts off by downloading the archaea parent dataset. It'll run BB tools first, and then it'll run prodigal on that, pass those results to Hammer, and it'll score it. So 20% it scores on archaea. Then it does the same for bacteria. It skips BB tools because it's already run, and then runs HMM search on the results there, and it scores 100% here. So it's looking promising for bacteria. Where have we gone? There. And then after that, it'll download eukaryota, and it'll do the same thing again. Mini-prot is the default here. So it's unlikely to be eukaryota because we know it scores 100% in bacteria, but we'll just wait and see what it produces, and then we'll see what it does next. In a couple of minutes. Well, it shouldn't take that long. see, so there's a little done there.
We use this time for a question. That's all right.
Yeah, sure. Yeah, if there's a question coming in, please.
Cool. For Metagenomics versus Augustus, how sensitive are they to teasing apart genomes for relatively related taxa? E.g., I'm working on lichen, which can contain multiple fungal bionts. Will I be able to tease apart BUSCO completeness for different bionts with Metagenomics, or would Augustus be preferred?
Okay. So normally those kind of biology questions I would pass to another member of the team who has probably more expertise in the biology side of things. I can if you the specifics of that question probably warrant a more complete answer than I can give at the moment. So if you want to post that question on the GitLab issue board, I can pass it to Mose or someone else in the team, and they can give you a more precise answer. Because I don't really have any more precise information other than what I've already said. That, like, I guess Augustus has more precise gene models, and Metagenomics is, you know, quicker but slightly less precise. So I mean, I can't really speak to the particular issue that you're talking about. But I can follow up with it if you just post a note on GitLab. I'll be happy to follow up.
That's fair enough. And then I have 47 samples from 6 closely related plant species. I would like to pull out the BUSCO loci and plot a phylogeny for them. What would be the best way of doing this?
Okay, yeah, I'll be talking a little bit about that later on in this talk, so hang tight. There's a section coming up on plotting trees, phylogenetic trees, and a couple of other use cases. So I'll be talking about that very soon. This is taking longer than it should, this run. Okay, I'm going to cancel this, and I'm just going to run it on actually a good way to show you an alternative way of running the auto-lineage pipeline here. Instead of auto-lineage, I'm running on archaea, bacteria, and eukaryota. You can run auto-lineage proc, and that just runs on the prokaryotic data sets, so archaea and bacteria. And similarly, you've got if you know you're already dealing with the eukaryote, you can skip the prodigal runs on the archaea and bacteria data sets. So here we're just going to do proc, and I'll change the output directory so it doesn't complain. Auto-lineage pipeline. So that'll just run the same things again, and it shouldn't take long this time because we already know that it's a bacterial genome, but I just want to show what happens after it figures out that it's bacteria. So, okay, so it's selected bacteria. And now it takes the markers from that it's matched a bacteria, and it's searching the tree for the chosen lineage to find the best taxonomic match. Here it's downloading a number of files, tree files that we also provide on the servers, and lists of reference markers for the bacteria dataset. And then it uses this information along with the markers that it has extracted to place the markers on the reference tree. So here now it's running SEP to try and identify which data set or which node on the tree is the most suitable for this input file. This might take another minute or two. So if there's any other questions coming in, feel free to ask them.
There is another one. Do we have the score for each best predicted gene according to Hammer in the output?
Say that again. Do we have
the score for each best predicted gene according to Hammer in the output?
Yes. So we have in the Hammer output file, which I'll look at after this is done. We can go into the results folder again. There's a Hammer output folder, where it has all the information for each of the markers and how the marker scores against the entire genome or the entire protein set. So you'll have information about the scores for each gene against each hammer profile, or each HMM profile at that point. So we'll look at that now. So it's identified here. It's run 7 and it's identified Enterobacterales as the most appropriate lineage, supported by 46 markers out of 47. So it's going to run this again, like the regular pipeline. And then we get the output score here. So it scores 100% in 100% completeness, 99.5% single copy, and 0.5% duplicated. So that's 2 genes are in multi-copy. This is quite a good result. So now we can have confidence that this assembly is a good quality assembly because we've chosen the most specific data set. So we'll look into the auto-lineage pipeline output here. So this is the folder here. So we've got the short summary files, and we've got the specific short summary file for the specific data set, and we've got the generic short summary file for the parent data set. We've got the run bacteria, and that's a hyperlink to another folder that's in the auto-lineage. So the auto-lineage directory contains the information for archaea and bacteria and eukaryota if you're running eukaryota as well. Then run Enterobacterales is where the main output is going to be. It's interesting output. So you're going to have a full table here, which you can use to identify which markers, for example, are duplicated. So this could be an interesting thing to look at. So this marker is duplicated, and it matches these 2 genes here. So the question was about the hammer output. So we'll look inside the hammer output here. And then the hammer output is here. So here we've got the output from HMM search for each marker gene. So we'll look inside one, for example, this one here. Okay, so this one just had 1 match. So you just got 1 gene here, and we got the score information for that gene. Try and find one that has more than 1. Let's pick another random one. No, that's the same. Yeah, I'll sort them by size. That's probably the best way to do it. That should be like this. That's the directory. Okay, so the biggest one, we'll look at one Enterobacterales, our output 63042. And this has lots of matches. So you can see these are the different genes that matched, and you got the score column here. You can see this one here scores a lot higher than the other ones. So it's likely that this is the only one that was accepted after all the filtering steps. Okay, so that's the auto-lineage pipeline. Let's see. Is there something else that I just want to bring to your attention? If you're working with fungi, the fungi data set, there is an extra file in the data set that contains a list of markers that are typically missing in parasitic reduced genomes. And if the missing BUSCOs that you have, say if you're running a fungal data set, a fungal genome, and you have a high missing score, and these missing markers match the pattern of a parasitic reduced genome, BUSCO will report this and give you an updated score as if these markers weren't included. So just something to take into account if you're working with fungi. So the BUSCO command line, there are a number of different options. We've seen a few of them already. The mandatory options, you need to have the input and the mode. So those are the 2 basic ones. Along with them, you're probably going to want to also include the number of threads or cores to use, the lineage data set, because if you don't include the lineage data set, it'll automatically trigger the auto-lineage pipeline, which isn't necessarily what you want. And then the output file, the output folder name, you can specify as well. You can also specify the output path, which is a separate command. So the name of the output folder shouldn't have any slashes or anything in it. It should just have the name of the output folder. If you want to save it in a different path location, then use the out path option. There are pipeline-specific options here as well. So the Augustus pipeline, it can be invoked using the Augustus command. Then we've got the E command, which sets the E value cutoff for BLAST searches. This is, I think it's set to 1 to the by 10 to the minus 3. You probably don't want to change that unless you really, unless you have a reason to. The limit is the number of BLAST candidate regions to pass to Augustus. That's set at 3, but you can increase that if you'd like, or change it whatever way you like. The long option, this invokes the Augustus. There's a number of Augustus scripts, and one of them is optimize the parameters. Running this Augustus script takes quite a long time. It can take a couple of days in some cases, but it can improve the Augustus results at the end of the day. So if you'd like to get the highest quality results possible, then you can run the long option. The Augustus parameters, these are parameters that you can pass directly through to Augustus. So anything that's listed in the Augustus documentation as possible parameters for Augustus, you can pass directly through to Augustus using double quotes, then minus parameter 1 equals value 1, separated by a commas. So a list of the values that you'd like. You don't need to do that for the species though, because Augustus species has its own separate command option. So just to show you a little, I'll just out of this for a second. Back to this. I want to see if I can find the Augustus config path. Right, so here we go. This is where Augustus information is kept. So we're going to have a look in here, and in here we've got a species folder. And these are all the species that Augustus uses. So you can specify a species model that you want to use to train your Augustus predictions. And in the data sets as well, while I'm here, we'll look at the BUSCO data sets. So we've got eukaryote data set here. In the dataset.cfg file, we've got this value here, species. So it specifies the species as fly. So that's the value that will be used unless you override it with another one of these that you want from the Augustus set. So the Metauke pipeline, it can be invoked using the Metauke command. And then similarly, you can pass Metauke parameters to using the double quotes and then the list of parameters and their values as follows. And you can, for the two different Metauke runs, you can change the values. Say, for example, the sensitivity. If you want to change the sensitivity from one run to the next, you can do that. I showed a plot earlier that showed the results of changing the sensitivity value. Currently, the sensitivity is set to 4.5 for the first Metauke run and set to 6 for the second Metauke run. They were the parameters that we decided were optimal based on our analysis, but they can be overridden using these commands here. The miniprot pipeline can be invoked with the --miniprot. It's the default already for eukaryote, but it's an option for prokaryote. So if you prefer to run miniprot on your prokaryotic genome instead of prodigal, you can do that using this command. For all the genome mode pipelines, you can skip bbtools. So if you've already got your assembly statistics somewhere else, you can skip that step. If you're running bbtools, you can also pass these two commands through to bbtools. They're directly commands from the bbtools documentation. And the scaffold composition writes the ACGTN content per scaffold to a file. And then the config break sets the number of contiguous ends that signifies a break between contigs. And the default value is 10. The other options that are available, the config file. I'm going to illustrate this, the config file. So where did we, was it exercise 3 where we downloaded BUSCO? Yeah, it was. So we're going to go into the exercise 3 BUSCO repository. Now this is the same as the, well, I'll show it here. I suppose it's easier on GitLab. we've got the BUSCO repository, easylab/BUSCO. We've got the config here and a config file. If you don't like adding options on the command line, instead you can use this config file. You can uncomment whatever lines you like and set them to whatever you want. And if you want, you can also set paths to different versions of tools if you really want. But this can be an alternative to adding commands on the command line. And then when you finally run it, you save your config file, and then you just run config and then the path to your config wherever it is. And then all the commands, all the parameters that are saved in this config file will be used in your BUSCO run instead of having to tack them all at the end. Okay. What else? Download dataset. Actually, I'll illustrate this as well because this is nice. So if we want to download, say, the fungi dataset we were talking about briefly, download fungi. You can just do it with the download command. So when you're running BUSCO, it will download it automatically. But if you want to download datasets in advance and then run BUSCO in offline mode, you can do it using the download command. So if we look inside the BUSCO downloads directory now, we've got fungi, bb10. And in here, we've got all the same files as normal, and then this extra one, which I was talking about just before, the missing and parasitic, where you can have a list of all the marker genes that are typically missing in parasitic reduced genomes. So that's the download command. The download base URL. This normally you shouldn't need to touch this. It's just if you want to get your data sets from a different server. Currently it's hard coded to our servers. You don't really need to worry about that. The download path option is to set a custom local location for downloaded files. So in all the examples we've been doing, they've been saved in a path that's called BUSCO downloads with a lineages subdirectory that contains all the data sets. But you can change that by setting this path. The force option overwrites an output directory with the same name, and the restart option restarts a previous incomplete run. So the restart mode can be useful if you're doing a long run and say something happens, I don't know, the system crashes or some step fails, say the hammer step fails. You don't have to go all the way back to the start. You just have to go back to the last checkpointed point. So checkpoints after every external tool call. So say once Augustus is complete, it'll checkpoint, so you don't have to repeat an Augustus run. If one step in the auto-lineage is complete, then you don't have to redo that step if it's already completed, and so on. List data sets. I'll illustrate this again. So BUSCO list data sets. And this will look at the file versions file to extract the most recent list of all the data sets available. So here we've got, well, scroll down. We've got all the possible data sets that you can choose. So for bacteria, there's something like 83 data sets. Here, archaea, there's 16, I think, and eukaryota, there's 67, and 27 viruses. This is all OrthoDB10 data sets. And I'll be talking about the update to this very soon. The offline mode allows you to just indicates that BUSCO shouldn't attempt to download the files. So if you've downloaded the files already, you can just say this is where the data sets are, and use the offline option to say don't try to download them again. Quiet mode suppresses all log messages, and the tar option compresses some of the output subdirectories, because some of the directories can be quite large. If there's a data set with thousands of markers, the HMM folder can be quite large, for example. And then the BUSCO output sequences is a sequence for each marker, so you can compress those directories as well using the tar option, just to save a bit of space. Generate plots. I'm going to illustrate this as well. So there's a companion, or there's a script that's part of the BUSCO software package. So if we go to, say, an output folder here, let's go to the Augustus genome pipeline. Actually, we'll just use that as the working directory. So we're going to use generate plot.py. And then, well, you can see the different options that are available for it here. You really only need this one option here, the working directory. So we're going to say the working directory is the Augustus genome pipeline output folder. And then it's going to read the short summary files to create a plot. So here we can have a look in here. So it's exercise 3, the Augustus genome pipeline. And there's this BUSCO figure. There we go. So we can see that we scored, most of them are missing. So there's 47 that were found out of 255. So it's just under 20%. It was like 18%, I think it was. So you can also stack a few of them together. So for example, if you want to, we'll just try this now. We'll make a directory for the short summary files, short summary. I haven't tested this, but I think it should work. And then we're going to copy the Augustus short summary.txt information into the short summary files. And then do the same for the Metagenomics short summary files. And for the miniprot. So now the short summary files should contain 3 different short summary files. Now I'll do the same thing again, generate plot.py, the working directory of the short summary files, and see if this will work now. So in exercise 3, we've got short summary. Where is it? I cannot see it.
You might need to refresh the folder.
Okay. Just by opening it again. Does that work?
Should do.
Refresh here. There we go. Ah, yeah. Okay.
Oh, there we go.
Okay, short summary files. Perfect. Okay. And here we've got 3 of them stacked on top of each other. This can be useful as well if you're comparing multiple runs. That's the generate plot script. So the next exercise will stay in Gitpod. It's the batch mode. So we'll go to exercise 5 here. So switch to exercise 5. Exercise was 5, yeah, exercise 5. So here we've got a folder that's already present. It's called batch input. And inside here we've got 2 genomes. So instead of passing a single input file, you can pass a folder instead of a single files. So we'll just copy this command here. I've got the batch input as your folder here instead of an individual genome file. The output we're going to call batch input pipeline. The mode is genome, and we're going to use the bacteria dataset again, just for illustration purposes. I'll make this bigger, we can see it. So here we've got the batch mode, the log looks a little bit different because it's going to run each of the files one after the other. So we got the 100% here, and then it goes on to the next file, and it'll do the same with the bacterial genome mode. Okay, so that's all done. So we'll look at the output here, batch input pipeline. And the output of the folder looks a little bit different now. We haven't got the short summary files at the top level. They're going to be at the next level down. So we'll go in here. We've got the logs file here, and we've got this batch summary file, which contains a table of all the input files that you ran and how they scored, the single score, duplicated, fragmented, missing, etc. And the genome information, the statistics, the N50 score, contigs N50, the percentage gaps, and the number of scaffolds. So if you're running hundreds of genomes, this can be a useful file to look at all of the output in one place. And then to get all the standard output, you just look inside these directories that are named after the input files. And you've got the same structure as before, the short summary files, the logs for all the external calls, prodigal output, etc. Okay. So that's batch mode. BUSCO use case examples. Okay, so this is, we're going to look at this protocols paper, which we published a few years ago. It's a very useful paper because it's got a number of different, a step-by-step approach to lots of different ways that you can use BUSCO. So we'll go to, there's a companion repository here, and we'll go to that in a second. And there are companion pipelines that can be used for visualizing syntonies between genes and for constructing phylogenetic trees. So this is the question that came up there just a few minutes ago. So as far as the way again, we'll go to, so this is the paper here. Go to the top. So this is the assessing genomic data quality and beyond. It's in current protocols. There are these protocols that are described in the paper. And I'm just going to look briefly at support protocol 2 and support protocol 3. So support protocol 2 is on page 24, I think. Around about, where are we now? A bit further up. Yeah, here. So visualizing syntonies between genomes using BUSCO markers. So there's a companion repository here. So we're going to copy this protocol repository. You can see it's here, this repository here. And there's, so support protocol 2 is here. And then there's a subdirectory for plotting syntonies and a snake file. So we'd be using snakemake to run that. So for example, if we just have to, let's go back to, next page, let's go. I'll just do it here. We're going to clone, we'll do it in exercises. I haven't set up a dedicated directory for it, but we'll do it here. Git clone this directory here. It's already cloned there. So it'll clone the protocol repository, which we were looking at. And inside this BUSCO protocol repository, you go to support protocol 2 and plot syntonies. And this is all explained in the paper. So we can see here, it shows you to go to this plot syntonies subdirectory. And you can find 2 full table TSV files, which we've talked about already, that show which markers are found, incomplete, duplicated, missing, or fragmented. And then you run the snakemake script, which is simply snakemake cors2 use conda if you like. And then the output is this kind of plot here. We're not going to do it now because it's the setting up the environment with all the different R packages just takes a little bit longer than we've got. But just so you're aware that this pipeline is well explained in this paper, and you can use it for your own analysis. Similarly, for building phylogenomic trees, there's another support protocol here in this directory, support protocol 3. And that similarly has got, where is it, rules, is it? The snakemake rules. The snake file is here, sorry. So you can just run it in that directory. So again, you follow the instructions here, you clone the repository, you go to the support protocol 3, you gather all your GFF files, and then so this is just stepping through all the process as an example, and run the snakemake script. And you can create your own phylogenetic tree. As part of this, BUSCO is run in protein mode, and the results are used to construct this tree. So this is worth digging into if you're interested in phylogenetic analysis using BUSCO. But yeah, so I'm just kind of highlighting it there, and I invite you to spend a bit of time looking into it later on. Also, it's possible to use BUSCO, the BUSCO results to train gene predictors as well. So we've got the results that we use BUSCO results to retrain Augustus, but it's also possible to retrain other gene predictors as well. Yeah, that's what you'd like to try and do. Okay, the final section of the talk, I'm going to talk about the data sets. So We've been promising updated data sets for a couple of years now, and we appreciate everyone's patience. But they are actually imminent. We have a paper accepted at Nucleic Acids Research, as I said, and that's just been accepted. So by the time that's published, we'll have all the data sets online and ready to go. So a little bit of information about them. The AutoDB version 12 update has a big increase in the species coverage. The current data sets that we have, the version 10 data sets, are from 2018 data. So you can see, compared to these numbers here, eukaryota has increased 4 or 5 times. Bacteria has increased right the way up to 17,500 species. And then archaea has also increased. So with this much larger species coverage, we can generate much more representative data sets that are less biased towards particular clades or subsets of species. So these are the figures that we've got. We're up to over 500 data sets now in the upcoming release. The AutoDB10, there was 193, including 27 virus data sets. And now with the big increase in, particularly in the bacterial coverage, we've got over 300 bacterial data sets. The eukaryota data sets, the final number is still to be determined, but it looks like it's around about 113. There might be a slight variation in that when it comes to the final publication, but all the same, it's that we've got a much higher number of data sets, and it should enable you to do your analysis at a much more high resolution, get better results for your assemblies. I'll talk briefly about the dataset creation procedure. The data selection, it's all data that's obtained from AutoDB, so the version 12. First of all, we run all the species against the parent data set from AutoDB10, and if there are any species that have high duplication, we remove them. Then we remove any species that score below either 90% complete or the clade median complete score, less 2 standard deviations. They're the filters that we apply to the species that we use. Then we propagate all species from leaf nodes up to parent nodes. So even if there was a species removed at one of the filtering stages, we'll include it in the parent if it passed the thresholds for the lower down clades. We discard any clades with fewer than 10 species remaining, because we want to have at least 10 species per data set, just to have a representative sample. After that, we take only the best scoring 100 species from each family. This is important for the clades higher up the phylogenetic tree. We don't want to be biased towards one particular family. And then we take only the best scoring 5 species from each genus for a similar reason. We want to kind of make sure that we're balancing the representation of all of these genera. So once we've selected all the data sets, all the data in the species data, we create an alignment of the genes within each orthologous group at each level using cluster-low. Then we trim the alignments with trim-all and build the HMM profiles with hammer build, HMM build. After that, we score. We run HMM search on the profiles and determine the appropriate threshold scores for the matches. And these are the threshold scores that we will include in the data sets. Finally, we run all the species in the clade against the data set and remove any markers that perform poorly. So if they have a low F1 score, like less than 80%, we'll remove those markers. And then we remove any data sets that are very similar to a parent. It happens in some cases that, say, a family or an order data set are very similar to each other. They might have exactly the same species composition or the same marker numbers, the same species numbers. So if they're very, very close to each other, we'll just keep the larger of the two. So a preview of how they perform. So these are 2 examples of data sets. And we can see that they perform quite well compared to AutoDB10 data sets. In some cases, you might see on these plots that there are some species that score better with the AutoDB10 data sets. And that could point to maybe those species, the AutoDB10 data set being slightly more biased towards that subset of species. And similarly, it could work the other way as well, that a data set might be biased against a certain subset of species, and they might score better on AutoDB11. So we've been doing a lot of this analysis in anticipation of the release, just to make sure that we're happy with the results. So this is the paper that's coming up, OrthoDB and BUSCO update. And so we've got information on the newest version of OrthoDB and the BUSCO updates as well. So keep an eye out for that. It's coming soon. And I think that's everything. So thank you for your attention. And if you have any questions, I'll do my best to answer or get back to you after the session. Yeah, thank you.
Cool. Thank you very much, Matthew. Do we have any questions? Well, actually, I have one. So with the parasitic gene notice, is that specific to fungi because it's a well-studied, well-known thing? For example, if I tried to run it on parasitic wasps, could I get the same notice, or is there just not enough information in BUSCO for that?
So at the moment, we just have the missing in parasitic file included in the fungi dataset.
Right.
Yeah, so it's not. I guess in the future we may expand it, but at the moment it's just fungi. Yeah.
Okay. Cool. Thank you. Right. Is there anything else, or should we let Matthew get on with his day? No? Cool. Right. Well, thank you all for attending, and thank you, Matthew, for giving up your time to give the session. It's been very interesting.
A pleasure. Thank you. Thank you for the invitation.
And welcome. And I guess I'll see you all in the next sessions tomorrow, which I think include wise ancestors. So that should be quite interesting to see. Right. Goodbye.
Bye-bye. Bye.